Hello, everyone, and welcome to our webinar today discussing the user experience and compliance in human microbiome research, specifically surrounding gut or fecal microbiome studies. My name is Laura Cunningham, and I am a microbiome product manager at DNA Genotek, and I'm delighted to be moderating this session. We are regularly asked for data to support the use of our stool collection and stabilization device. First-time users are worried about their customers or study participants and want reassurance that the stool collection process is as simple and efficient as possible. So today, we will be discussing some important considerations and some challenges and solutions to ensure you are maximizing your budget and making the stool collection process as pleasant as possible for both you and your donors. The agenda for today's webinar is as follows. I will take care of introducing you all to the interactive webinar platform. I will then introduce you to our two speakers who will then present their respective talks. Following the presentations, we will have some short poll questions, and then we'll finish off the session with some general Q&A and discussion. So before we begin, I wanted to highlight a few tools on the webinar dashboard. At the bottom of your screen, you will notice multiple application engagement tools, which you can resize, move, or close. You can expand your slide area or maximize it to full screen by clicking on the arrows in the top right-hand corner. Please submit any questions during the webinar during the Q&A engagement tool highlighted here on the slide. We will try to address as many of your questions as possible during the Q&A portion. Additional content to complement the session is available in the related content section marked with a paperclip. Lastly, an on-demand recording will be available and sent out approximately 1 day after the webinar. The two speakers presenting today are Dr. Crawford Parks and Melanie Abrahamson-Sommer. Dr. Crawford Parks is an R&D scientist at DNA Genotek working on the product development team. Tara joined in 2019 and currently leads multiple projects to support the verification and validation efforts of our molecular product portfolio. Prior to DNA Genotek, Tara received her undergraduate degree in biomedical science with a minor in chemistry and her PhD in cellular and molecular medicine from the University of Ottawa. Melanie Abrahamson-Sommer joined the Oregon Health and Science University's Department of Endocrinology Bone and Mineral Unit in 2006 and works as the department's project manager on their observational and clinical research studies. Melanie also serves as the lead program manager for several clinical studies, including the Mr. Oz research study and several rare bone disease studies. The Mr. Oz project is a multi-site study of osteoporosis in older men that started in the year 2000 and recruited 5,994 subjects, which we'll be learning more about later in today's webinar. Welcome, Tara. Welcome, Melanie. Thank you for joining me today. And without further ado, I'm going to pass things over to Tara for the first part of the webinar.
Thank you, Laura, for your introduction. As mentioned, today I will be discussing data that we have collected to evaluate the usability and user comprehension of the OmniGene Gut product. OmniGene Gut is our all-in-one system for easy self-collection and stabilization of microbial DNA from feces for gut microbiome profiling analysis. This product can be easily used to self-collect a high-quality sample in the comfort of one's own home, which paired with an intuitive and user-friendly collection method ultimately improves donor compliance and provides standardized fecal samples ready for transport, storage, and downstream processing. Today, I will provide an overview of data we have collected to evaluate the usability and overall user comprehension of the OmniGene Gut device. Specifically, I will discuss a first study, which I have separated into two parts for simplicity, Study 1A and Study 1B, that was comprised of both an observed study using simulated fecal samples, as well as an at-home mail-in study where participants collected real fecal samples. The second study I will touch on was done in partnership with the Crohn's and Colitis Canada and was designed to evaluate the usability and performance of the OmniGene Gut device in a targeted dysbiotic population. First, to provide an overview of the first arm of Study 1, our observed study, which was conducted using simulated fecal samples, was designed to evaluate user compliance and IFU comprehension. The use of simulated samples here allowed for direct observation and provided a means to evaluate if users could correctly perform the critical tasks from the OmniGene Gut instructions for use. These simulated samples were developed to mimic real-world sample types across the Bristol scale and are composed of recipes containing peanut butter, oats, liquid, and other grain-type material. For this study, we targeted 120 participants split across 4 user groups, comprised of naive users representing the general population. The user groups were based on education level and age. As you can see here, we had 2 education levels, education above high school or GED and below. In addition, the age categories which we targeted were age ranges from 18 to 44 years of age or 45 years and older. An observation checklist was provided to the observer and was based on the critical tasks of the IFU. An image of the IFU provided with the device is shown on the right-hand side of the slide here, with the identified critical tasks for our study as the following. Open the tube correctly, collect the fecal sample, transfer the fecal sample, remove the excess fecal sample from the tube, close the tube, mix the collected sample, and ensure the collected sample is properly mixed. In this study, the observer noted any incorrect actions or difficulties experienced by the user during the collection, with all critical use errors, close calls, and operational difficulties noted, and root causes were explored during the post-collection interviews. Endpoints that we evaluated from this study included the comprehension questionnaire success rate, as well as the success rate of the observed critical tasks, data which I will highlight in combination with Study 1B results in a few slides. In this study, it is important to note that our final study population successfully met our target of representing the general population, with a total of 118 participants and equal representation across user groups as well as gender. In the second arm of Study 1, we evaluated user compliance and comprehension of the IFU when used in an at-home environment. Importantly, this study was also set up to assess the ability of naive participants representing the general population to mail back study materials via General Postal Services. In this arm, we targeted 30 participants split across 2 user groups based on education level, which are the same education levels that we had in Study 1A, education above high school, and high school or GED graduate and below. Study participants received an OM200 device at their home. They collected a fecal sample into the OM200 device following the IFU. Participants answered a questionnaire and then followed instructions provided to mail back the samples using General Postal Services. The samples were then received at our processing lab and visually inspected and DNA extracted. Similarly to Study 1A, the study endpoints included comprehension questionnaire success rates, as well as collection compliance, which in this study involved visual inspection at the lab, as well as downstream DNA yields. Similarly to the simulated sample arm of this study, the final study population for the at-home mail-in study had good representation across age and education levels, as well as gender. In this study, we also asked participants to self-report where their fecal samples fell within the Bristol scale. And as shown here, the majority of the samples were type 3 to 5, which is expected in a generally healthy population, with some participants reporting type 1 and 2 or type 6 and 7. Across this large 2-part study, we had a total of 152 responses to the comprehension questionnaire that asked specific questions regarding the critical steps of the IFU. From these responses, participants answered the questions correctly 94% of the time. Success rates showed no correlation across user groups that were based on age and education, indicating that the IFU is easy to comprehend for the general population. When assessing the user compliance of the OmniGeneGut IFU, as evaluated in Study 1B, 97% of participants complied with collection instructions, whereby sufficient DNA was obtained per extraction aliquot to support common downstream microbiome processing applications. Across both the studies, 100% of study participants complied with instructions whereby the tube was securely closed. 97% of participants complied with collection instructions regarding sample mixing as assessed via visual inspection, either by the observer in Study 1A or by the laboratory personnel in Study 1B. The next study I will focus on was done in partnership with Crohn's and Colitis Canada, where untrained participants were recruited through their system to target a dysbiotic population. The purpose of this study was to evaluate the compatibility and performance of the OmniGene Gut device for in-home self-collection by participants undergoing Crohn's and chronic colitis symptoms. The main objectives were to evaluate performance of the OM200 device in lower diversity sample types and to also evaluate the user comprehension and compliance of the product IFU. There were 25 naive participants in this study that were provided the standard OM200 kit and also our OM-AC2 spoon accessory to aid in collection of a more liquid sample type, which is shown here on the right-hand side of the slide. Following collection, participants completed a comprehension questionnaire similar to the other studies, composed of questions related to critical steps of the IFU, and samples were returned to the collection coordinator and then provided to the laboratory for downstream processing. In this study, we had 65% of participants self-report their fecal sample type as a Bristol type 6 or 7, indicating more liquid sample types, with those who reported type 7 showing a preference for the OM-AC2 spoon accessory for their collection. The remaining participants who provided a sample of Bristol type 6 or below reported a preference for the standard spatula that's provided in the OM200 kit. Across all participants, regardless of the collection tool selected, there was sufficient amount of stool return in the OmniGene Gut device to support the DNA extraction method, which required a minimum of 50 milligrams of stool per 350 microliter extraction aliquots. In addition, the average DNA yield per extraction aliquot was sufficient for processing on common downstream microbiome sequencing platforms such as 16S and metagenomic sequencing. Taken together, the key takeaways from these usability and user comprehension studies demonstrate that the OmniGene Gut device is easy to use across the general population, including untrained, naive users, thus maximizing compliance for studies conducted using this device and minimizing failed collections. We have fully validated the user instructions for naive users collecting sample types across the Bristol scale. The OmniGene Gut device provides a user experience that is geared towards removing common barriers with fecal sample collection, and participant feedback was collected from Study 1A and B that indicated a willingness of users to collect from multiple kits from the same fecal sample, as well as recollecting at a future time. Finally, these studies provide strong evidence to support success of this product for at-home self-collections across the general population, which positions OmniGene Gut for scalability and increased reach for various study cohorts, while also highlighting its potential use in a direct-to-consumer environment. I hope that I have provided you with a comprehensive overview of our usability and user comprehension data for the OmniGene Gut device. And with that, I will pass it over to Melanie, who will walk you through a case study that will further highlight the ease of use for this product.
All right. Thank you. So I'm excited to present today about the osteoporotic fractures in men's study, Mr. Oz, and the user experience and compliance in human microbiome studies. So a little overview of Mr. Oz. This is a research study funded by the NIH. It began in 2000, so 21 years ago, which is crazy to think about a research study lasting that long. It is a multi-site longitudinal observational study of older men. We have 6 sites representing different parts of the US. We have Oregon and California on the West Coast. We have Pennsylvania on the East Coast. We have Minnesota to the north and Alabama to the south. All subjects were enrolled at baseline, so not adding any additional subjects throughout the years. Osteoporosis has often been associated with older women, but men also face increased risk of bone loss as they age. Prior to Mr. Oz, there was a lack of information concerning osteoporosis in older men. The deficit of information made it difficult to develop methods to diagnose, assess, and prevent male osteoporosis. Mr. Oz was established to identify the risk factors associated with osteoporosis and bone fractures in older men. So when originally we recruited the men, they had to be 65 years old or older in 2000. They had to be ambulatory, live near a clinical site during the study period, able to adhere to study procedures, which is pretty standard in every research study, and have no bilateral hip replacement. We wanted to be able to get a bone density scan on at least one good hip and hopefully follow that, follow the changes in that hip throughout the years. So we have lots of longitudinal data. For the main Mr. Oz study, there have been 5 clinic visits. The visits are spaced out about 5 years apart. We are currently seeing participants for visit 5 at least one of the sites. There were some delays due to the COVID pandemic, and I'm sure everyone has experienced delays in the past year due to that. But visit 5 started in August of 2020 and is, again, we're currently seeing participants. We have frequent contact with the subjects. 3 times a year, we send them a 1-page questionnaire asking about falls, fractures, pain, mobility. We also ask them if they have plans to change their address. The older generation tend to move around a lot. They move out of their houses into retirement homes, assisted living facilities, with family. So we want to keep tabs on them and update their contact information as needed. So why collect stool? I asked the PI this question, and he said the microbiome has very important effects on a variety of biological functions that may be very important for musculoskeletal aging. There are few studies of the gut microbiome and its relationship to aging and to the musculoskeletal system. Mr. Oz is a large study of aging men in which we have lots of information about musculoskeletal health. And assessing the gut microbiome offered a unique opportunity to greatly increase our understanding of these interactions. The study collected stool samples at clinic visit 4 and clinic visit 5. Again, which is currently taking place. The mean age of the participants at visit 4 in 2014 to 2016 was 85 years old, and it took place at all 6 clinical sites in the US. For visit 5, we wanted longitudinal data, so subjects had to have completed stool collection at visit 4 to be eligible to collect at visit 5. And the mean age of participants at the current visit 5 is 88.8 years. And it's taking place at 3 of the clinical sites due to some limited funding. So we chose the Omnigene OMR 200 microbiome collection kit. This kit is very user-friendly. There are detailed instructions with pictures walking the participants through the collection process step by step. It also includes the collection tube and a small spatula to use for collecting a very small amount of stool for the tube. Again, the instructions are step-by-step and include pictures. This is a great help when the subjects take the kits home to collect the samples. If someone needs to assist them with the collection, like a spouse or a caregiver due to their age, these instructions are really easy to interpret. Another option is the all components box. This includes the paper toilet accessory, a biohazard bag, an absorbent material, and a box for mailing. At our site, we had extra supplies from other studies, so we chose to purchase just the collection kit itself. Some modifications for the older population. We chose to use a plastic hat versus the flushable paper toilet accessory. We just thought this would make it easier for the men with hand tremors or arthritis in their hands or even vision problems. Simply placing the plastic hat on the toilet and giving the sample. The flushable paper toilet accessory, on the other hand, is a great option. It's easy to use. It's very small with literally no cleanup. After you use it, you simply unstick it from the toilet, wait 1 minute, and flush it. Very easy, very small, easy to mail it to people if needed. So we definitely have an advantage by having face-to-face visits with the subjects and giving them the collection kits in person. At the clinic visit, we have a collection tube that we use to demonstrate the steps of collection. We screw off the purple cap on the top. We demonstrate using the spatula to collect a small amount of stool. And I usually tell them it's about the size of the pinky fingernail, just a little bit of stool. Then we, pretend like we're swiping the stool with a spatula on the top of the yellow tube, which deposits it into the container. We then screw the purple top back on and demonstrate the shaking of the sample for 30 seconds. Again, just that hands-on, in-person interaction. We also enlarge the directions and include our clinic phone number so that the participants can call us if they have any questions. A few people, they spilled it or something, so they can just call us and we would send them another sample kit. So mailing completed kits back to the clinic. We provided postage paid envelopes to make it easier on the participants. They were able to drop the samples off in their outgoing mail. There was no special delivery needed. There's a special sticker that is placed on the back of the envelopes that's required by our postal service. And I have a picture of those on some upcoming slides. We also provided a biohazard bag, cotton balls, alcohol wipes, and paperwork, in which I'll go over in a little more detail in a few slides. So we chose to use the 6x9 bubble mailer. This added some cushion to protect the stool samples from harm when going through the mail system. To date, we have not had any samples damaged in the mail, which is great. We added return address labels and prepaid postage on the envelopes for the participant, again, making it as easy as possible for them. Our campus mailroom weighed the package for us and gave us a postage price. This helped us nail down the budget beforehand. We were also able to purchase the postage in our mailroom, and the costs were charged directly to our research grant. So again, finding those upfront costs for budgets was really helpful. All right, here is the special sticker that has to go on the back of the envelope. It's a requirement of the US Postal Service. So I'm sure it's different in Canada, but this is what we had to do in the US. We gave the men biohazard bags and cotton balls for the absorbent material in case of spills. And an alcohol wipe was given also just in case there was any residual stool on the outside of the tube. We just asked them to wipe off the outside before sending it. So the study wanted to know how long it would take for the samples to travel door to door, the participant's home to our lab. We had one lab at the Portland site that received all of the samples from all the sites across the country. We constructed this collection worksheet that includes the time and the date the sample was collected, as well as the date mailed. And then when our lab received the samples, they would note the quality of the sample and the date the sample was processed. I think the recommended room temperature time of the sample to stay stable was 30 days when we first started collection in 2014, but has since increased to 60 days, which I think is still the case today, stable at room temperature, 60 days. And then I'll talk about the quality of the samples in a few slides. So the study wanted to collect data about user experience. So we designed this questionnaire asking 4 simple questions. Would you volunteer again for this type of collection? Were the instructions easy to follow? How easy or difficult was the collection process on a scale of 0 to 10? And how long did the collection process take? We also left room for comments, and I have the results from this towards the end of my presentation. I wanted to note that we did compensate our subjects, but not for the stool collection itself, for the entire 4 to 5 hour clinic visit. The subjects had multiple scans, they had blood and urine collection, physical measures, and questionnaires. We gave the participants the gift card at the end of the visit, but noting that the stool collection kits were sent home with the subjects, so they received the money before producing the stool sample. So I don't think the compensation necessarily swayed one way or the other, whether they would collect or not collect. Okay, so visit 4 collection. Again, the mean age for the visit was 85 years, and this was in 2014 to 2016. The men who completed the visit, we had about 1,300 people. Of those men, 982 agreed to collect a sample, 290 refused, and 57 were deemed ineligible. Some subjects who came into the clinic might have had some cognitive impairment, mobility issues, arthritic hands. We just kind of used our best judgment, and in some cases, we just decided against giving them the collection kits, and those are the deemed ineligible. Some participants agreed to collect a sample, but never returned one, and that is 31. So of the 982 that agreed to provide a sample, we received 951 from visit 4. Of those, 6 were unusable, and that was, leaked in the package on the way here, or maybe a completely dry sample. So something happened to that, whether the liquid spilled or whatever the case, but 6 were unusable. So we ended up with a total of 946 usable samples. At visit 4, we didn't have a cleanly constructed database for tracking the sample quality, but we took detailed notes and have that information for everyone, just not in a clean database. Some comments were thick sample, dry sample, low volume, leaky sample. We wanted to know the condition of the samples, so down the road when we went to analyze them, we could look back at our notes, and if anything looked funny, we could maybe correlate with that to maybe a collection issue. So at visit 5, again, the mean age of this visit is currently 88.8 years old. We have the 3 sites collecting the samples, and there are 310 participants eligible for this visit. Of those so far, we've received 116 samples, 10 have refused, and 2 were deemed ineligible. And I think both of those were because of cognitive issues. There are 182 potential samples that we could still receive, and our hopes are to get around 70% of those. All right, so for the visit, we do have a nice and clean database for sample quality at visit 5. We have a check all that apply option. We have correct amount of stool in the tube. So out of the 116 so far, we have 79. More stool than needed in the tube, dry sample, stool leaked in the envelope. We had a couple of those. The metal ball was removed that you shake to help it mix. I don't know how that was removed, but it was. We just, again, want to know everything we can about these samples. We also had a few low volume or very little liquid. Just to note, we do get good samples from the more stool than needed and most of the dry or little liquid samples. Again, we just want to know, again, as much as possible as we can about the sample quality. All right, so this questionnaire was on an earlier slide. We want to know, how did it go? At visit 4, we started by only asking one site to complete this questionnaire, and that was the Portland site. At visit 5, all 3 sites are currently capturing this. Collected samples, visit 4, we had 174 from Portland only, and visit 5, we have 116 so far. Completed the questionnaire, we have 157, and visit 5, we have 105. So again, the questions were, would you volunteer for this kind of collection process again? We've got, let's see, 87% at visit 4 said yes. And at visit 5, we have 79%. Were the instructions easy to understand? Visit 4 was 97% yes, and visit 5 is 95% yes. On a scale from 0 to 10, how easy or difficult was the collection process? At visit 4, the average was 2.2, and at visit 5, the average was 3. Lastly, the average time to collect the sample at visit 4 was 9 minutes, and at visit 5, it was 9.5 minutes. So pretty similar answers at both visits. Again, the people that collected at visit 4 also collected at visit 5, so they had done this before. In conclusion, we've had success collecting microbiome specimen using the OMR 200 microbiome kits. The study finds this collection very feasible in the older population. It's accepted by participants. The quality of the DNA derived from the samples collected at visit 4 was excellent. And we also have published a manuscript about successful collection in a population of older men, which is attached if you're interested in more information. And with that, I say thank you.
Awesome. Thank you again, Tara and Melanie, for your wonderful presentations. And apologies for our technical glitch at the start of the webinar session today. So before starting the Q&A portion, and thank you for those who have submitted your questions, please keep them coming. We are going to release just 2 short poll questions just to engage with our listeners. So we're just getting ready to launch the first poll question. So what are the most common participant barriers to stool sample collection and specimen return? So we're giving around 20 seconds. For you to respond, and then we'll take a look at the results. Okay, so you should be able to see here the results. Looks like concerns around hygiene and contamination and storage and handling of the sample are 2 of the higher barriers. Makes a lot of sense to me. Melanie, Tara, I'm sure you agree with this, but if there's anything to add, feel free.
Yeah, I think that aligns with my understanding of it as well. Sorry, Melanie, to cut you off there.
No, that's okay.
Aligns with what I would expect for this type of poll question.
Yeah, definitely. I agree with that.
Okay, awesome. Perfect. Thank you, everyone. And we'll release the second poll question now. So when choosing a stool collection device for your study, how important is compliance and usability data? So scale of 1 to 10, 10 being the most important. Okay. I think we're probably ready to release results. Gonna push the results to the audience here. Sorry. Oh. I'm not seeing, sorry, I think we're having issues with this one. I don't see, having some technical issues with this one. So we're gonna leave this question for now. And we're gonna get to start addressing some of the questions that have been submitted. So there's quite a few, so bear with us. First question for you, Tara. For the at-home study, were users provided additional information to coach them through any difficulties?
Sure, that's a good question. So for our study, we were really looking to get feedback and usability data from a naive population who had never seen our kit, never seen the IFU. So they were not provided any additional information. The only information they were provided outside of the IFU was instructions to follow on shipping it back once they collected their sample, to send back their sample in the General Postal Service, which is really key information that we gained from this study to support that, a naive user from the general population is able to follow the instructions and get it back to a processing lab sufficiently.
Awesome, thanks. And so Melanie, next question for you. Did your study collect information on food intake and medications the subjects were taking at the time of collection?
Yes. So we had everyone who collected a stool sample, we had them complete the block brief food questionnaire, the FFQ. The brief one is 7 pages long, and it asks, what participants have been eating for the past year. So a little bit of everything. We also asked them to collect the samples 24 to 48 hours after the clinic visit. And if they had eaten anything unusual that they normally don't eat, we asked them to wait 7 days before they collected. And also medications. We asked what they were taking, prescription and supplements within 30 days. So we knew exactly what they were ingesting.
Okay, perfect. Next question for you, Tara. And there's been a few questions I see, so I'm kind of hopefully compiling some to address one sort of underlying question. It's what additional considerations, if any, across the studies you presented were required when targeting a population with a type 6-7 Bristol stool sample, so more liquid stool?
Yeah, that's another great question. So as we highlighted in both our observed study and in the study 2, which was in collaboration with Crohn's and colitis, where the majority of people collecting had type 6-7 fecal samples, we provided in both studies our spoon accessory in addition to the spatula that's provided with the traditional kit. And we also gave the option for a plastic-based toilet accessory rather than the paper. So similar to what Melanie's study showed in her slide where she had the two side by side. So for a liquid type sample, we find that participants have an easier time collecting in a plastic-based toilet accessory rather than the paper one. So those were two things that we considered and provided to people in those study cohorts.
Thank you. I feel like the next question, both of you can, offer your 2 cents. The question is, how can we increase sample reliability using kits for home collection by participants?
I can start off by answering this. I think communication with the participants. So if that's a study coordinator or something, calling the participants just to check in, see how it went, even like before they collect the sample, calling them one more time before at their house. Do you have any questions? Let me know what you need. I think communication with the participant or the person collecting.
Yeah, I would echo that. Particularly for our at-home study, we also did something similar, confirming that the participants received the kits and reiterating the time frame that we need it back. And, if you needed any support or had any questions, they knew that they could reach out to the study coordinator.
Yeah. Awesome. That's great. So moving on to the next one. Have you studied compliance in older age groups and those with disabilities? As many diseases affect, people over the age of 60 years of age, it'd be good to know how this age group performs using the kits.
I can provide an answer, and I'm sure, Melanie, you have some commentary to add as well, just based on your study. So as mentioned, for our study, we really targeted the general population, and we were targeting, in that upper age range, 45 years and older. That captured, well above the 60 age range. And we were able to really demonstrate in that data that our instructions for use are simple enough to follow, and the collection was feasible in that age group, which I agree with you is very important because, as you mentioned, a lot of these diseases occur in that aged population. So our data just supports that the collection instructions are simple enough to use by those people.
Yeah, I agree. The instructions are pictures. Everything is so clear, and a lot of our older participants, when they take the kits home, sometimes they need help from a spouse or a caregiver or someone, and we really haven't had too many issues with other people helping. By looking at the instructions, they're very easy to follow. And we also, one other thing, we also give them our phone number, just so, again, if someone else needs to help them or if they have any questions, they can call one of us, a coordinator, just to ask any questions.
Yeah, that's great. So the next question comes from a listener who participated in the American Gut Project. And they said they used toilet paper to collect the stool sample and then used a swab for the fecal collection. And the comment is more, they don't think this is a great way to collect. Do you have any comments surrounding this? So Tara might, I guess, get you to answer this question.
Sure. Yeah, I can respond to that. So there's a lot of different formats of stool collectors out there on the market. And I think, like you said, maybe the method that you followed isn't the best method for getting sufficient or consistent samples. So I think what stands out for our OmniGene Gut product is, it's a volumetric input that is consistent. So across our studies, we see consistent yields for DNA yields from the product. It's reproducible. We have a lot of validation data that shows that collections from the same stool sample into multiple kits in parallel are consistent, as well as taking multiple aliquots from the same collected tube show consistency, which really speaks to the homogenization of the sample. So I think that those are all things that give, more confidence in the reproducibility and validity of the data that you would be looking to get from the collected stool samples.
Yeah, agreed. I'll just add, form factor is so critical for donor experience, but also from the laboratory and processing side as well. We might not have to think about, extracting the sample from a swab, but having, a liquid sample all ready to go makes the laboratory work a lot more seamless. Okay, this is great. The next question, feel like we addressed, but just in case there's anything extra, it's a congratulations for a very successful study. Any suggestions to further improve sample return and sample quality? Anything else you guys want to address here?
Again, I know that I have said this a few times, but I think the communication piece is just so huge with that. I mean, we had such a successful collection, but again, my study, these men have been in the study with us for 21 years. So we do have a very good relationship with them, but communication, just calling and checking in, sending the sample back. Are you going to send it back? What questions do you have for us? That would, make you feel a little bit more comfortable sending the sample back.
Yeah.
Yeah, I agree with Melanie on that.
Okay, perfect. Next question. If the study design allowed it, would you recommend collecting 2 samples per participant to ensure good DNA yield?
I can answer that. So the DNA yield from our study 1B, which was the at-home study, as well as from the study with the Crohn's and colitis cohort, we showed that we had more than enough DNA yield from a single aliquot. So that's just one aliquot from the full tube. So there'd be more to go back to. So from a DNA yield perspective, it might not be required. However, depending on the downstream processing, where that processing is occurring, you might need multiple tubes, and we actually collected information from our usability cohort if they would be willing to collect into a second tube, just to see their willingness. And we had very positive response rates from our donor cohorts, indicating that it would be no problem for them to collect into a second tube in parallel. So that's good information to have if you are looking for multiple tubes to be collected.
Yeah, I agree. I've done a few kind of just naive user calls as well, and it was something I always asked too. And the question hands down was like, yes, I would collect a second tube. You know, it's the most daunting is that first collection, but like once you're in there and once you're comfortable and you've done it before, it's not a big deal. That's the feedback that I've received as well, which is in line with what you just said, Tara.
Yeah, and I think it really lines up with the what the end goal at the laboratory processing side is. So if you what amount of sample you require for your downstream processing.
Yeah, and I'm going to piggyback on that. I think, you know, when we collected the samples, I think that we had a company analyze them and aliquot them, and I think they received minimum of 4 aliquots per tube. And I think that, you know, we even could have gotten more of that. So we had them aliquot, you know, 4, and then we had them send the whole tube back. So we have tons of collection material.
Mm-hmm. The next question actually ties in really nicely to this question, and it's, you know, can we use the samples, so OmniGene gut samples, for short-chain fatty acid analysis? So currently our OmniGene gut device is validated for microbial DNA. So short-chain fatty acids would be off-label. And we actually have recently released a new device, so same form factor as OmniGene gut, but specific for the stabilization of metabolites. So in this instance, you know, if someone's interested in collecting microbial DNA and metabolites such as short-chain fatty acids, you know, we would encourage the 2-tube solution in terms of collection. So collecting 2 tubes from the donor from the same stool sample. So hopefully that addresses that question. And again, it kind of brings back the point where, you know, 2 tubes is possible, again, depending, as per Tara, on the question that you're trying to answer. Okay, awesome. And then last question, just conscious of time here. Again, it's more looking for an opinion. They're wondering if, you know, sending a nurse to collect the stool sample would be a good idea. Do you guys have any opinions on that?
I think that sounds expensive, but if you, I read further, I read further down, and it looks like they were also collecting blood at the same time. So in that case, you know, if you have a nurse that's available, that's going into the home, I mean, certainly having that person there for the collection process is great. And I'm sure the quality of the sample would be, you know, great as well. I don't think it's necessary, but I mean, kudos if you can do that. That's great. Even a coordinator. I know that we did a few home collections, but we also always sent 2 people just for safety for, you know, going into someone's home. But yeah, I mean, if you have a nurse available, great.
Yeah, and just echoing off of that. So I think what Melanie said is correct. Like if you're also trying to pair it with blood draws, for example, to look at analytes both in the blood and the stool sample and use that in your analysis, then you would need to have those paired collections. But is it necessary from a stool sample quality aspect? I think our data really supports that people are able, from the general population, first-time users, to use this kit at home and provide sufficient material for downstream microbiome processing, so that it isn't required to have a professional nurse, for example, coming into the home.
Yeah, awesome. Couldn't agree more. And just so you all know, we fixed the technical glitch with that last poll question. So I'll show those results really, really quickly, and then I'll let you all get back to your day. So in terms of how important compliance and usability data is, 8, 9, and 10, 10 and 8 being the highest percentage. So I think, like, you know, people at DNA Genotek would be happy to see these types of results. You know, Tara's presentation really shows the extensive validation efforts that go into these types of devices. So happy to see that the scientific community also agrees with us on that. And with that, I just again like to thank our 2 wonderful speakers for taking the time to present today. Lots of great questions. So thank you to our listeners for being so engaged. And if you're interested in connecting with us, please feel free to do so. Otherwise, have a great rest of the day. Thank you.
Thanks, Laura.